Welcome to STRAS. So what do you mean by technology? Technology is nothing but something which can make your life simpler. Biotechnology is nothing but using biology to make your life simpler. So biotechnology is a subject which uses biology to make your lives simpler, our lives simpler. So when you speak about physics or chemistry, you'll be telling that atom is the basic or fundamental thing. Every is a basic or fundamental thing in biology. Cell is the basic or fundamental unit of it is, and cell is also called a structural and functional unit of life. Why structural unit? Because it will be making every structure in our body or in any organism. For example, in human beings, so it will give structure to heart, eyes, nose, legs, and hands, whatever the things are there. And functional unit because it performs the functions like digestion, reproduction, eyesight. We need cones and rod cells in the eyes to perform specific functions. And it also helps to respond to stimuli. Nothing but if anything come near your eyes will automatically close. So that is called response to stimuli. These all things are there or coded in something called cell. Okay, to become a cell, we need to have need to have a three characteristics. First thing is the cell will be having the cell or plasma membrane, which is called boundary of a cell. So cell will be having a boundary called cell or plasma membrane, and it will be having a fluid inside it in which all the organelles will be floating, and transport of different amino acids or molecules will be happening through this fluid, and that fluid is called cytoplasm. And it must have a genetic material, whether it is a DNA or RNA. We will be looking at about this DNA or RNA in more detail. Then features of living organisms. So any living organism, the features of them is they should reproduce, they should grow, and they'll be having a metabolism regarding digesting the food or nutrition to produce energy, to walk, to eat, to sit, to think. We need some energy. So for that, metabolism will be there for digesting the food and responding to environmental stimulus. So these are the things which a living organism does. These are the functions of the living organisms. So all these functions will be happening at cellular level. So cell is capable of independent existence. This statement can be asked in the prelims examination. So basic thing is cell is structural and functional unit of life, and cell can all the functions are happening at cellular level. So cell is capable of independent existence. For example, bacteria. Then we have different types of organisms called unicellular, multicellular. So only Monera and Protista are unicellular organisms. So unicellular will be having independent existence, performing all the essential functions of life: reproduction, growth, metabolism, responding to environmental stimuli. So anything less than complete structure of cell do not ensure independent living. So if they cannot do any of this function, they cannot live independently. And first cell was discovered, live cell was discovered by Antonio van Leeuwenhoek, and Robert Brown is the person who have discovered the nucleus. And the cell theory was given by Scleden and Swan, and Scleden and Swan have given two statements. This might be probably important for prelims examination. All living organisms are composed of cells and are made up of products of cells. And also all cells arise from pre-existing cells. So now let us look in deep into the cell. I mean, like what cell has and what are the organelles or what are the parts of the cell. Right. So cell will be having a cell membrane. So first, which I told that it need to have a cell membrane or plasma membrane, and it need to have a fluid called cytoplasm. Fine, and it should have a genetic material called DNA or RNA, and it needs to be organized. If it is organized well, then it is called eukaryotic. If it is not organized well, then it is called prokaryotic. Karyon means nucleus. Karyon means nucleus. Pro means old. U means new or true. Pro means old or false. This is a Latin word. So we'll be looking at all these organelles. So you might have studied in school mitochondria. It is the most important blank or most important question, 1 mark question that will be there. But here we'll be looking why it is called powerhouse of the cell. Then there will be centrioles. So it will be helping for cell division, vacuole for storage. So if you remember, just remember what are its function also, it will be very much enough because you are not going to write neat. Okay, mitochondria, powerhouse of the cell, centrioles, cell division, vacuole, storage, lysosomes for digestion of proteins, digestion of waste material. It means it is a recycle bin. It will be recycling the waste materials or the genetic materials which are waste debris which are present in the cell will be digested by lysosome. So just remember LD. Then ribosomes, which are present on rough endoplasmic reticulum. There will be 2 types of endoplasmic reticulums. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. So you can see. So this is the rough endoplasmic reticulum and this is the smooth endoplasmic reticulum. On the rough endoplasmic reticulum, we'll be having something called ribosomes. On smooth endoplasmic reticulum, we won't be having any ribosomes and ribosomes. Okay, so on the function of smooth endoplasmic reticulum is lipid synthesis. And the function of rough endoplasmic reticulum is protein synthesis with the help of ribosomes. Ribosomes are those which make proteins, or it is also called as protein synthesis, and they are helpful in the process called translation. We'll be looking at in more detail. Then Golgi complex. So it is used for processing, packaging, and distributing the lipids and proteins. So whatever the proteins made with the help of translation, with the help of ribosomes and the smooth endoplasmic reticulum, they will be processed in Golgi complex and they'll be packed by them and it will be distributing. Okay. Then we have rough and smooth. I have told about you about that. Cytoplasm is a medium in which transporting of materials will happen to one place to another place. Mitochondria to Golgi complex, nucleus to mitochondria. These things all will be happening in cytoplasm and every cell organelles will be floating in this cytoplasm. Then I told that U means true, carry-on means nucleus, eukaryon, true nucleus, before, prokaryon, cells underdeveloped or ancient or old cell, and eukaryon will be a fully developed cell. When it comes to organelles, specific chemical reactions or tasks are made by specific organelles. I told that centrioles, cell division, right? Ribosome, protein synthesis, vacuole for storage. So energy generation, mitochondria, it is called powerhouse of the cell, and it does aerobic respiration. It does something called aerobic respiration. Then chloroplast, it is present only in plants. So it convert food, it makes food from sunlight in plants. Vacuole for storage of cellular products. Then endoplasmic reticulum for transport and synthesis. Ribosomes for making of proteins or synthesis of proteins. Lysosomes, I told that digestion, it is a recycle bin. But recycling of waste will happen in lysosomes. Golgi bodies for packaging. Then nucleus, it is also called as brain of the cell. So another word for nucleus is carry-on. So this nucleus is called brain of the cell. Why it is called brain of the cell? Because it control all the operations or functions of the cell. So to control those operations, it have 2 components. One is information. It is packed, it is stored in a DNA material or genetic material called DNA or RNA. And these operations are used to control actual or specific commands. So there will be selective commands will be given at certain time. For example, a baby to be born. So first what happens is, so body should be grown. So in that eyes should be grown, then nose, then stomach, hands, legs. So these specific commands will be given by this nucleus. at certain point of time. So for that information is required. This information will be in DNA or the genetic material. Well, so now let us understand the genetic material DNA. So ribose means, so full form of DNA is deoxyribose nucleic acid. So ribose means a sugar with 5 carbon molecules. You can see that here it is a sugar with 5 carbon molecules. 5 carbon molecules. Then deoxyribose without any oxygen. Deoxy means without any oxygen. It means that it has no OH group in it. Then when it comes to nucleic acid, so nucleic acid is made up of something called nucleotides or nitrogen based compounds and phosphate groups. So P and N, nitrogen based groups and phosphate based groups, these are called nucleotides, which are there in DNA. You should just remember P and N, phosphates and nitrogen based compounds are called nucleotides. And this nitrogen based compounds are of are 5, generally 5. So one will be there in RNA synthesis called uracil. So there are 2 types, purines and pyrimidines. Purines are double bond, pyrimidines are triple bond. So adenine and guanine are purines, are nothing but nitrogen groups. Cytosine and thiamine and uracil are pyrimidines, which are triple bond in nature. I mean, like adenine will only tie up with thiamine and guanine will only tie up with cytosine. Okay. So this is DNA. DNA is a double helical structure. So when it comes to DNA, so we can see here deoxyribose, we can see 4, 5 carbons and 1 phosphate group, and we have something called nitrogen groups, and nitrogen groups in which we have thiamine, adenine, guanine and cytosine. So A will only bond with T, G will only bond with C, C will only bond with G, T will only bond with A. And this is the screenshot which is taken in class 11 in CRT. So A and G have similar structure and T and C have similar structure, but A will bond only with T and G will bond only with C. So just remember at the girls club. So to just remember this, at the girls club. So G will only bond with C, A will only bond with T, T only will bond with A, C only will bond with G. So deoxy means without oxygen. Nucleus means made of nucleotides, and it is acidic in nature. It has genetic information for cell growth, metabolism, reproduction, and response to stimuli. These things. So structure of DNA is double helix. So we have in EG purines and cytosine, thiamine are pyrimidines. So DNA stores information. So DNA stores information. So how does it work? So cell has a cytoplasm. In cytoplasm, we know that chemical reactions take place. Cytoplasm is nothing but the fluid in which all the cell organelles will be floating. So chemical reactions will be helpful for the functions of the living organisms, for us to see, for us to think, for us to walk, for us to eat, for us to digest. We need some chemical reactions to happen in our cell. So those information will be stored in DNA. For example, glucose plus oxygen. We intake glucose in the form of food, and through respiration we intake oxygen. So it will be converted into carbon dioxide in water. This is the specific reaction which happens in our body. So to happen this reaction, we need something called amino acids or proteins. These amino acids or protein, which protein should be released? That is the work, or that is the information which will be there in our DNA. So A plus B will leads to C plus D. To start this reaction, we need one catalyst called enzyme. So this catalyst is not a part of reaction, but it will start the reaction. And nucleus will decide when the reaction will start or when the enzymes will get made. And DNA has complete info of all these enzymes. So enzymes are nothing but catalysts to start the reactions. Nucleus will know when to start. And this information will be there in DNA. So DNA is the actual commander, and it has certain enzymes, and it will control all the functions of the cell. It will control all the functions in the living organism. And 99% of these enzymes are made up of something called amino acids or proteins, which are called building blocks of the organism. But all proteins are not enzymes. And all enzymes are proteins. This can be asked in prelims. So all proteins are not enzymes, but all enzymes are proteins. So enzymes are made up of amino acids or proteins which are there in DNA. The information to synthesis these proteins or amino acids, when, where, how, it is there in nucleus. And ribosomes make or synthesize proteins. The only thing the ribosome can synthesis is a single string. But we know that DNA is a double helical structure, double string. For this, we need something called a single string called RNA. So for the process to convert DNA into RNA, this process is called transcription. And from RNA to protein, ribosome will convert it, or ribosome will synthesis it, and this process is called translation. C comes first, L comes later. Okay, DNA to RNA transcription. RNA to proteins will be done by ribosomes. Ribosomes can synthesis only single string or single structure called RNA. So the process is called translation, and this proteins or enzymes will be controlling all the cellular reactions. And this is called DNA to RNA, RNA to protein is called central dogma of cellular genetics. So we will end up here, and I'll be teaching more about this in the next lecture. Thanks for watching. If you find it helpful, please do like, share and subscribe.